Hello friends, welcome to my channel Teach Guru. Today we will talk about related to DNA replication. I convert DNA replication in three different video. First one is Meselson and Stahl experiment. Second one is DNA replication mechanism. How to DNA replicate in this second video. Then third one is modes of DNA replication. So we will start our first video. Meselson and Stahl experiment. First of all, 1953, Watson and Crick give a molecular structure of DNA. Let's give a molecular structure of DNA, but not true. DNA replicates by semi-conservative mode, conservative modes, or dispersed modes. Then 1957, Meselson and Stahl, let's conduct experiment and prove that semi-conservative modes of DNA replication play important role for DNA replication. Third one. Now these particular slides are Meselson and Stahl figure. Then three types of mechanisms are involved in replication, but DNA replicates by only the one mechanism. So I explained this the three mechanism in next slide. This particular slides. Semi-conservative modes of DNA replication. First one, that's double-stranded DNA are replicate, and one strand are old strand, and second one is newly synthesized strands are joined. That second one, conservative modes. Two strands are replicate old strands, and two strands are newly synthesized strands. Then dispersed modes of DNA replication. Dispersed modes are some parts are newly synthesized in this particular DNA strands. Some parts are old synthesized, some parts are newly synthesized. This difference between these three types of mechanism. Then start this. DNA are made up of nucleotides. Nucleotides are made up of nucleosides and phosphoric acid. Nucleosides are made up of pentose sugars and nitrogen base. This experiments main importance according to this experiment and nitrogen base. And source are used this experiment and 40 and 50. Okay, and 40 are it's called light nitrogen and 50 as heavy nitrogen. This particular experiment that used in the particular experiment, in bacterial strain are used. Bacteria are used and grow in E. coli. That's bacteria are E. coli grow on petri plates and this contain and 50 containing medium. And 50 containing medium and isolate from DNA from this medium and include in enter in cesium chloride medium and mix it and give band in bottom sides of beaker. Okay, and that's DNA are this particular DNA. And then second generation, E. coli used from first generation. And this contain and 40 containing medium and isolate from DNA and enter in cesium chloride and mix it and then bands are give, and 40 and 50 hybrid DNA are observed in this particular second and 40 and 15. And then third one generation is used in E. coli from second generation bacteria. Then mix it and 40 containing medium and from isolate from this medium DNA and then give a band, particular bands. Okay, that's give a 4 different types of bands, 4 different types of DNA. 2 strands are and 14 because this use in the bacteria in from second generation and 40 containing medium bacteria. Then 2 strands are made up of 40 and 50 hybrid strands of DNA. So this. Okay, so to prove that DNA replications by semi-conservative modes, let's prove that in this particular experiment. And this particular experiment, this conduct that the DNA replication are either by Meselson and Stahl experiment and the DNA replication by semi-conservative mode. Okay, thank you friends. I hope you like this video you like this my video, and you please my channel subscribe and like that and give me comment in particular any video. Okay, thank you.